Hello students, welcome to today's class. I am Bernisha, lecturer from Emmanuel College of Pharmacy. Today we are going to see about percolation and oscillation techniques used in the extraction of crude drugs. In the last video we have learned about the extraction techniques which is used in the extraction of crude drugs from the plant and animal tissues. Today we are going to see a detailed view of percolation and oscillation. Okay, let's get into the video. First, let's see what is percolation. Percolation, it is known as the continuous downward displacement of the solvent through the bed of crude drug material to get the extract. It is fully like an continuous downward displacement with the of solvent. So here the solvent is continuously poured over the bed of crude drug so that at last we will get the extract of that crude drug. Here mainly it is used to extract or active ingredient mainly in the preparation of tinctures and the fluid extracts. It is mainly known it is the method of short successive maceration. Here let's see what type of instrument is used for this percolation method. Here percolator is used for the percolation technique. The percolator is a narrow cone shaped vessel with open at both the ends. We have a wide open at the top and the small lid like opening at the end. These are the percolators which is used for the percolation techniques. Here there are two types of two shape of percolators are used. Most commonly it is used in the industries and somewhat it is also used in the laboratories in the colleges for the percolation process. Here, I have already said that these percolators have a two openings and it is also a narrow cone-shaped instrument. So, in the top of the percolator, there is a wide opening through which we can keep our crude drug or herbal powders which is used for the extraction. And the below or the bottom of the percolators there is a small drain valve which is used for the draining of our extractum which is present in the percolator after the extraction from the crude drug. Here within the picture we can easily identify where we will keep our herbs or the crude drug for extractum. Here we already said that the top of the layer there is a wide opening and below and the bottom there is a drain valve. In between there is a perforator plate inside. So in the perforator plate means nothing but the plate which has some of the holes which is used for the um, extracted liquid, that is our menstruum can easily passes through this liquid, passes through this holes and get into the drain valve so that we can easily drain the extractor from the percolators. In olden days they have used some of the filter papers or washed sands, so that in modern techniques they have used the perforator plates, but in the olden days they have used the filter paper or washed sands um, for the extraction process. And after that while uh, passing through that the menstruum will pass through this washed sands and filter paper, then it reaches the drug powders, and with the help of the glass wool we can easily separate the impurities which is present in the extractum, and with the help of the drain valve we can easily collect the extractum which is which contains the complete active ingredients which is soaked after soaking it from the herbs or crude drugs. So these are the percolators which is normally used in our laboratories and industries. Next is steps involved in percolation. For the percolation process, there is a size reduction. First, there will be the size reduction, so that the drug which is to be extracted, there it has to be size reduced so that we can easily get extracted. So usually we have to convert the coarse powder drug into the fine powder drug. Next, imbibition. Imbibition is nothing but moistening of the drug product. So we first, before soaking the drug product into the solvent, we have to completely moisten the amount of drug which is to be kept in the closed containers for few hours before getting into the percolators. So during imbibition, our powder drug is moistened with the suitable amount of what type of menstruum we are using, and it has to be stand for few hours in a well closed container. So first is size reduction, and the next one is imbibition, and the next one is packing. After the imbibition, the moistened drug is packed into the percolator. So I have already said that above the perforated plate, we have to keep our drug which is moistened using the menstruum. And the next is maceration. Maceration is the soaking process. So here the drug is completely soaked in the menstruum. So after packing, we have to add the sufficient amount of menstruum, and it has to be stand for more over 24 hours to get the pure active ingredient loaded extract. So next is percolation. Next we will collect, so that the lower tap, the drain valve should be opened a little bit, so that the extract will collect by drip slowly. Drop wise it will be collect, so it may be around 3/4 of the value. If we have collected 3/4 value of the menstruum, then we have understood that, so that we can easily collect the remaining menstruum from it. To till until it reaches the 3/4 of the volume, we have to allow the drain valve to drip the extracts. So we have to control the rate and control the flow rate of the menstruum. So these are the five steps which is involved in the percolation process. First is size reduction, and the next is imbibition, next is packing, next one is maceration, and again the percolation. This percolation, there are many types of percolations. The first one is simple percolation. Here we have already said that the same process will be continued. First we have to take our drug, and we have to imbibition for 4 hours, then maceration for the 24 hours, then percolation we have to collect the percolate. This is the simple process as the steps involved in the normal percolation process. So it is known as simple percolation process. For example, tincture of belladonna, compound tincture of cardamom preparations. So the here simple percolation means it involves the steps which is used for the normal percolation process. And the next one is modified percolation. Modified percolation means repeated maceration, which is more effective than the simple percolations. Here we have to soak it repeatedly. So first, in the simple percolation, we have to macerate the drug only for 24 hours. But in modified percolation, we have to repeat the maceration process. Here the solvent we have to we are going to add in that is the menstruum. We are going to, for the extraction process, has to be divided into equal multiple time considering the solvent retained by the plant tissues. Here mainly it is used to prepare the concentrated extractions. Mainly the concentrated solutions are prepared using this modified percolation. This also maintains the steps involved in the normal simple percolation, but there will be the repeated maceration process. And the next one is reserved percolation. Reserved percolation means this is the type of simple percolation, but there will be some of the extra procedures, like there will be an evaporation process is there. So the equipments like climbing evaporator will be there for some the evaporation process, so that we will get a soft extract like semi-solid type of products. We will get the semi-solid type of extracts. With the help of this clamping evaporator, the extra water present in that extracts will be removed, so that we will get a soft or semi-solid extract. This is known as reserved percolations. It also shows the steps involved in the normal percolation process. So all the three types of these percolations, which involves the simple percolation steps, but in modified percolations we have to repeat the maceration process, and in reserved percolation we have to use the clamping evaporator to get the consistency of the soft extract. Here, next one is the merits and demerits of this reserved percolations. Here, mainly the reserved portions, that is the semi-solid portions, will be strongly alcoholic and easily dissolves than the evaporated portions. So, there will be some of the risk for precipitations. So, as it is in semi-solid conditions, so and it has the alcohol, so it is that when the alcohol get easily evaporated or easily dissolves, the portions remaining will undergo some precipitations. The merits means it requires a less time than maceration process, because we does not need to stand for 24 hours or 48 hours. It requires less time than the maceration process. And extraction of the thermolabile constituents. The substances, the constituents which are very sensitive to the heat may also easily extracted using this process. And the demerits, it requires more time than the saucillation process. So when it's compared to the saucillation process, it requires more time and more solvent is also required, and a very skilled person who knows to one who knows to um, how to handle, one who knows to how to handle that evaporator, so that we have to keep a skilled person to handle this reserved percolation process. These are the some of the merits and remerits of the reserved percolation process. And next one is saucelate extractions. We have studied the percolation process. What are the types of percolation process, along with the steps involved in the percolation process. Next, we are going to learn about the saucelate extraction, which is also known as saucillation process. So here we have to use a saucelate apparatus. In this picture, we can easily identify this is the structure. This is the construction of the saucelate apparatus. We will keep the round bottom flask on the mantle, and there we have to keep the solvent. And next, we have to then, like percolator, we have to imbibition. There it may undergoes imbibition, then packing of the drug powders, then there will be the condensation process, then extraction takes place. So these are the some of the main parts of saucillate apparatus. Here the saucillate extraction is the continuous process of extractions with mainly the hot organic solvent. So here we have to use the solvent continuously. Typically this saucillate extraction mainly used to have limited solvent, which of the solvent, which of the drug which has the limited amount of solubility in the given solvent, we have to use this saucillate extraction process. And we have mainly we have to add, so we have to unmonitored and unmonitored efficiently, it will recycle the small amount of solvent. Mainly we have to add the small amount of solvent in it, it continuously evaporation then condensation, so that there will be manageable, so it can easily manage itself. So we don't want to get monitored or managed the saucillate extraction. So once we have filled the solvent, it's get recycled and we will get a large amount of extraction. From this picture, we can identify the saucillate apparatus also. So saucillate apparatus is mainly used for the saucillate extraction. This is used for the drug products which is has limited solubility in the solvent, and the impurities which is present in the drugs is insoluble in the solvent. Within this both the conditions, we are using this saucillate extraction. Next is saucillate extractor. We have in the last slide we have seen the saucillate extractor, which is invented by Franz von Saucillette. Hence, it is named by the saucillate extractor. So it has three main sections. First is the percolator. It contains a boilers and reflex. It mainly circulates the solvent. The solvent gets circulated in this percolator. And the next one is thimble. Thimble is nothing but where we will pack the drug powders, which is usually made up of a thick filter paper or cotton, which will keep the drug powders or crude drug which is to be extracted. And the next one is siphon mechanism, which is mainly periodically empties the thimble. In the picture itself, we can see these three problems. One is percolator, then is thimble, and another one is siphon. First is thimble. Thimble is the main source. There means where we will keep the extracted crude drug. The crude drug which is to be extracted will be kept in the thimble. It is the main chamber of the oxalate extractor. Mainly here, the percolator. Inside the percolator, we have to keep our crude drug. That particular loaded part is known as thimble. And the distillation flask or round bottom flask, which contains the extraction solvent, which is used for the extractions. Heating mantle, which is required for the maintaining the temperature, and there will be the reflex condenser which is placed at the top of the extractor, which contains the which allows the inlet and outlet of the water, and it which helps in the condensation process, so that the vapors get easily converted into the water droplets. Next is water inlet and outlet. This reflex will contains the water inlet and outlet, through which the water gets easily get inside the condenser, and then it goes outside the condenser through the outlet. With the help of this cool water, there will be the process of condensation will occur continuously. The working procedure. I have already said that here the bottom of the extractor has a round bottom flask which will have the solvent. So the round bottom will have the solvent. That green color is our solvent, and the above the part is known as the percolator. That is the percolator part. So inside the percolator there is a green color matter. The green color particular matter is known as thimble. So inside the green color there is a red color particle. So the red color particle is known as our crude drug. So outside there is a U-shaped or curve. That curve will be the packed thimble. So the drug will be easily packed in the inside the thimble. And the next one is there is a small U and some of tube-like structure in the near the percolator area. That tube is known as chiffon tube. With the help of the chiffon tube only, the drugs which gets extracted, the constituents which is extracted from the crude drugs will be passed through the chiffon tube, and then it is collected on the solvent flask. And the next one is above the percolator there is a condenser, through which where we will get where the water gets in inlet, and there is a water outlet. So what is the working procedure? First we have to set up the extractor. First is our round bottom flask, then we have to keep our percolator, then above that we have to fix our reflex condenser. So first in the round bottom flask we have to take the solvent. So the round bottom flask will be kept on the heating mantle. So some temperature, a suitable temperature will be given to this condenser. So while giving the temperature, the solvents get boiled. So at a certain temperature, it attains the evaporation state. While the solvent gets evaporated, the vapors will passes through the um, tube. So here the red color arrow mark shows the way of evaporation. So the way of vapors get into the condenser. So here with the help of this tube, through the red color arrow, the vapors get passes through the condenser. While once it reaches the condenser, with the help of this water inlet, the vapor. So the vapor gets converted into water droplets. Once the vapor enters into the condenser, it gets cooled and the vapors will be converted into the droplets. So the droplets are getting down into the thimble. So thimble contains our crude drug. Once the droplets get fully enters into the thimble, once it reaches the certain level, it collects. So once it reaches the above the thimble, the tube, the chiffon tube collects the drug, and once it reaches the above level, it then continuously comes down into the round bottom flask. So with the help of this red color arrow, the vapor process and get into the condenser. There the vapor will cool and there undergoes condensation process. Once the drug gets truly soaked or macerated, the siphon tube will collect the extract. Once it reaches the above place, once it reaches certain place or that bend, the extract will be collected on the solvent flask. And again this procedure repeated until our crude drugs get completely extracted. So only the small amount of the extract is needed for this oscillate apparatus extraction. So continuous hot extraction. So we don't want to manage, or no, we no need to change the solvent, or no need, we have no need to add the extra solvent for this continuous hot extraction. So it is the simple it is a simple one of the simple extraction process, but it shows more, which helps to collect the more amount of extract which is used for the collection of extract from the crude drugs. Here we have some of the working procedure. That is, first we have to keep the solid materials in the thimble, and which is the oscillate extractor will be placed onto the flask containing the extraction solvent. The oscillate equipped with the condenser. The solvent is heated to reflux. The solvent vapor travels up to a distillation arm. This arm is known as the distillation arm, and which enters into the thimble of the solid. The solid material will definitely fills with the warm solvent, where the vapors get into the condenser and there it cools and they are converted into droplets, and that droplets will slowly fills the thimble with the solvent. The desired compound dissolves in the warm solvent. Once the oscillate chamber is almost full, the chamber is emptied by the chiffon tube. The solvent running back down into the distillation flask. This process continues until we have reached the full time of extraction. So this is the working procedure of the oscillate extraction. And again, this cycle may allow to repeat for many times, over hours or days. During each cycle, certain portion of the compound dissolves in the solvent. After many cycles, around 72 hours, the desired compound is concentrated in the distillation flask. After the extraction, the solvent is removed, typically by means of rotary evaporator, yielding the extracted compound, so that the extract compound will also have the solvents, so that which will be removed with the help of the rotary evaporator, like resolved percolation process, and the non-soluble portion of the extracted solid remains in the thimble, and which is usually we will discard that non-soluble portions. So this mind map which will be helpful in the understanding the cycles. First we have to keep the powder sample, and we have to put the sample in the thimble, place the thimble in the extractor, fill the solvent in the round bottom flask, heat the oscillate extraction unit, reflects the solvent, and there will be the extractor. So this is the simple mind map which will be useful for us to understand the oscillate extraction process. And the next one is the advantages of this oscillate extractions. So mainly it is mechanically gentle on the samples, but still efficient in separations. It is when the we are gentle. This type of mechanism is gentle on the samples, but it is very useful for the separation process, and it is able to extract the solute which is insoluble in the which has the insoluble impurities, and it is often used as a benchmark when developing new extraction methods. It allows for unmonitored and unmanaged operation. I have already said that we have no need to change the solvent materials, or we have no need to change the simple or like that we don't want to and monitor the full operations. It is the most useful apparatus for solid liquid extraction in various fields such as pharmaceutics, environment, foodstuffs, etc. These are some of the greatest advantages of the continuous hot extraction. And the next will be the disadvantages. What will be the disadvantages means there will be the long operation time required so that it may reach some of the several hours or days, and may be sometimes there will be the large solvent volumes are required. We have to add the large volume solvents in the round bottom flask for the first time itself, and the evaporation and concentration needed at the end of the extraction. At the end of the extraction we have to undergoes the evaporation and concentration of the extracts, and the inadequacy of the thermolabile analyze. There will be some of the inadequate conditions for the thermolabile analyze. So these are some of the disadvantages of this oscillate apparatus. So in this slides, we have learned about the oscillation process along with the continuous percolation process. In the percolation process, we have learned about um, what is percolation, what are the steps which is involved in the percolation, and what are the types of percolations which is used in the extraction of crude drugs. And next one is the oscillation process. Here we have completely learned about the oscillate apparatus, the procedure, working procedure of the oscillate apparatus, along with the advantages and disadvantages. Okay, students, I hope you all have learned about the percolation and oscillation process. Let's see in the next video. Thank you.